In this lecture, we're going to talk about bacterial reproduction and how microbiologists measure the growth of microbial cultures. Before we do that, though, I'd like to take a minute to connect this lecture with our metabolism unit by talking about just how important all of the catabolic machinery of the cell is in order to allow something like reproduction, which requires a ton of anabolic processes in order to make twice as much of everything inside of the cell, so that when reproduction occurs and the daughter cells are formed, each of those cells has all of the things inside it that it will need to survive and thrive. So basically, the ATP requirements of the cell that we discuss in the metabolism unit are necessary for growth and reproduction. So growth, in the case of microorganisms, we're going to find in this chapter, is not growing larger so much as it is growing and dividing and creating offspring in a process that typically involves the cell dividing into two genetically identical daughter cells, and because they divide into two and they split, that process is called binary fission. In addition, prior to that binary fission, the manufacture of all of the cellular components that are required for each of those cells is made, and that utilizes ATP. And when binary fission or asexual reproduction isn't occurring, the cell is using ATP to repair cellular components because of that process of entropy, which you talk about in general biology, where everything sort of tends toward chaos. There's a constant input of energy needed to keep that cell functioning well. So I want to just take a moment to relate what has to happen in order for binary fission to occur and talk about how much that costs in terms of ATP. So first of all, when you're undergoing binary fission, you need to replicate the chromosome. So in a typical bacterial cell, the chromosome is about 3 million nucleotides. Notice here that in order to replicate DNA for every base pair you add of those 3 million, there is a requirement for 10 ATP, which means to replicate the chromosome takes about 30 million ATP. So no small amount of catabolism is required in order to just make the DNA. In addition, the cell needs to make all of the proteins that will both become the structure of that new cell. Think of something like peptidoglycan, and all of the enzymes that that cell is going to need in order to do all of the jobs that the cell needs to do. And in order to transcribe each of those proteins, you have an ATP requirement of about You have an ATP requirement of about 5 ATP per nucleotide. Translation costs about 3 ATP or more per amino acid that's added to the growing polypeptide. And if we look specifically at cell wall synthesis, which is going to be a big part of binary fission because the cell elongates to about twice its size and then divides, it takes about 4 ATP per sugar portion of that peptidoglycan. So you have the requirement for transcription and translation of each of those proteins, and you have 4 ATP required for each of the sugars. So it's energetically an incredibly expensive process to undergo binary fission, but it's the only way to get your genetic information passed on to the next generation. And so it's a cost that bacteria are willing to shoulder. Now, though there are a couple of different ways that prokaryotes replicate, we're going to talk specifically about something called binary fission, which I mentioned earlier, which is where the cell undergoes this process of elongation, replication of the cellular components, and then division into two completely distinct cells. Now, this, like many things, is something that is easier to understand when it's happening dynamically, when you're watching the process occur. So I'd like you to take a moment to watch how this process occurs and what it looks like in the cell. Now that you understand how binary fission occurs, I want to talk about something called biotic potential. And biotic potential is not something that's specific to microorganisms. Any organism that's capable of reproduction has a biotic potential, the potential to grow and populate a given area. The biotic potential of any organism is dependent on two major things. The first is how many offspring you have with each reproductive. The first is how many offspring you have with each. The first is how many offspring you have with each reproductive event. And the second is how quickly you're able to make that reproductive event happen. So in the case of binary fission, the number of offspring is always doubled from the parent number. So one cell becomes two cells, and there's no changing of that. There's no multiple offspring. If you think about something that has multiple offspring, think about a rabbit who has multiple babies per litter, right? So that's an increase in biotic potential because of the number of offspring, and that does not occur in bacteria. Bacteria always undergo binary fission, so there's a doubling, but there isn't any increase beyond that. However, microorganisms, bacteria and archaea that undergo binary fission can have an incredible biotic potential because they are able to reproduce so quickly. They are able to move from one binary fission event to the other very, very quickly. And so their biotic potential has nothing to do with the number of offspring they have per reproductive event. It has everything to do with how quickly they can move from one reproductive event, go through all of the rigmarole of making all of the pieces that are necessary, and then undergoing another binary fission event. And just as a point of reference, Clostridium perfringens, which we're going to see in a few moments, is a wound pathogen, can undergo binary fission every 6 minutes, which is crazy when you think about the amount of energy that needs to be stockpiled and used to make all of those pieces, and then to actually go through the process of binary fission. That can happen every 6 minutes means that Clostridium perfringens has insane biotic potential. Now, to look at something that's a little more routine, something like E. coli has a generation time of about 20 minutes. Now, that may seem like a lot less compared to Clostridium perfringens, but if an organism is capable of reproducing every 20 minutes, that means every hour they can undergo 3 rounds of binary fission, which means they can undergo 20 rounds of binary fission in less than 7 hours. And what that means is that that cell can grow and divide and become over 1 million cells in that 7 hours. So one cell can become 1 million in less than 7 hours, and that, my friends, is a huge amount of biotic potential. So again, the biotic potential of microorganisms is not attributable to the number of the number of offspring per reproductive event. So the biotic potential of prokaryotes is not due to the fact that they can have multiple offspring during a reproduction event. The biotic potential of microorganisms is due to the fact that they undergo these binary fission reproductive events so very quickly. And that's not true for all microorganisms. Something like Mycobacterium tuberculosis takes about 15 to 20 hours to replicate, and there are some environmental bacteria who take 60, 80 hours in order to replicate. So there are some very slow growing microorganisms, and we would simply say that they lack the biotic potential of those that can undergo binary fission more quickly. Regardless, we attribute the biotic potential of microorganisms to their rapid generation time. To talk about something called a biofilm, which is a really cool thing. Well, it depends on where it's growing. The electron micrograph at the bottom left-hand side of your screen is a biofilm that's growing on a catheter, and it has Staphylococcus aureus embedded in it, and that means that this person has a very serious infection. So that's not a cool thing from the human perspective, but from the perspective of the microorganism, the ability to produce something called a biofilm is a really cool thing to be able to do, because it allows the cells to exist in this highly protected environment where they can share nutrients and remove wastes and even plan and coordinate reproduction. And from our perspective, it's very bad because it allows those microorganisms to resist antibiotics and all sorts of things that we're going to talk about when we talk about trying to control microbial growth. So a biofilm happens when microbial cells land on some surface, and that could be the rim of a bucket, it could be a urinary catheter, it could be a prosthetic joint, and that attachment, because they produce chemicals, happens very, very quickly, and then they become irreversibly attached to that surface fairly quickly, and then they undergo binary fission and grow and grow and grow and grow, and then they produce this EPS and form water channels. So they're almost like this city of microorganisms, and they grow across the surface of things, and then as they grow, they can actually release new microorganisms to disperse off into new sites in the patient or on the bucket that's holding the water sample, or in your drains of your sinks, or in the ducting of your air conditioning. And they're really, really nasty little things in terms of human infection, because they make it very, very difficult for us to remove microorganisms and treat bacterial infections. And like I said with the last slide, the biofilm, again, is one of those things that I think is helpful to see happen dynamically. So I'm going to add a little video here so you can see that.
The life cycle begins when bacteria enter the bloodstream from various infection routes and evade the immune system or antibiotics. Formation begins with attachment of planktonic bacteria to the surface, preconditioned with proteins from the host immune system. Irreversible attachment to host proteins signals the bacteria to create biofilm and is the first committed step of biofilm growth. The bacteria anchor themselves permanently using cell adhesion structures such as pili and receptors called epitopes. This encourages new bacteria to attach to newly arriving cells, providing adhesion sites and building the protective matrix of the biofilm structure. Cells now communicate via quorum sensing and use small molecules to regulate gene expression within the bacterium. The community grows through both cell division and recruitment. The development of this complex biofilm makes the community resistant to the immune response and antibiotics. It may include diverse species of bacteria, yeasts, minerals, and host proteins. Recruited neutrophils result in a cascade of inflammatory cytokines and host proteases. These damage tissue and regulate fluid accumulation and exudate, providing nutrients. Bacteria now exist in multiple forms and metabolic states. Now a different phenotype, both physically and metabolically, it has the survival advantages of diversity and multiple defenses. The life cycle continues. as fragments or planktonic bacteria are released to reinfect the human host.
Bacterial growth, we are going to be talking about all of the ways that we use to control the growth of microorganisms based on what we know about them from our study of them, which we talked about in the last lecture. Before we get going, though, I want to talk about a couple of different terms, and this particular table came from the Tortora textbook. There is a similar table in the OpenStax textbook, which gives us a couple of different terms that I want us to be familiar with before we move into our discussion of the control of microbes. First up is the word sterilization, which is probably familiar to you, but just that we have a context for what we mean when we say sterilized in this class. We mean the destruction or removal of all forms of microbial life, including endospores. So endospores are the hardest thing for us to kill, and so that's what we aim those techniques at. The one exception to the way that we typically sterilize would be those prions, the non-living proteins that we talked about in the first lecture unit, which require really intense levels of chemical and physical control in order to destroy them. So sterilization is aimed at killing endospores, and when they kill the endospores, they kill everything beneath that, including viruses and all of the living microorganisms. Another word that may sound familiar to you and may be one that you use in your common language is disinfection, but from a microbiological standpoint, the word disinfection is a word that means to destroy pathogens on inert surfaces or fomites or inanimate objects. So any of those three words, inanimate, inert, or fomite, mean a non-living surface. And anytime we clean to remove microorganisms from a non-living surface, we call that disinfection. When we are attempting to remove those microbes from skin or mucous membranes, we call that antisepsis. So the difference between antisepsis and disinfection, notice, is just whether or not that thing is safe for use on skin and mucous membranes. So a lot of times disinfectants, say like bleach or something like that, are very effective at killing microbes, but they are very effective at damaging us as well, so we don't use those as antiseptics. So disinfectants are for use on inanimate objects or fomites. Antiseptics are for use on skin and mucous membranes. Sort of a follow-up term to antisepsis is something called degerming. Degerming is the removal of microbes from a limited area. So a really common example of degerming would be to wipe down the skin with an alcohol wipe prior to giving an injection or taking a blood sample from a patient. And in that case, you might use something that would typically be considered a little too harsh to be used as an antiseptic, like alcohol, but because you're using it in a limited area, it is considered that the benefits of that outweigh the possible harm to the skin. So degerming is sort of a special form of antisepsis where you're removing microorganisms from a limited area of the body. Finally, we have the term sanitization. So to sanitize is to lower the number of microbes in a given area, and this is usually applied to food service, but we also sanitize areas in the lab, and we do that in order to lower the number of microbes to a safe level. All right, now that we've got that terminology down, I want to talk a little bit about how we control microbial growth, because it turns out that controlling microbial growth doesn't always mean killing microbes. Sometimes it just means to inhibit them from growing. So we have a couple of terms that we use generally. We use them and they describe for us the way that a particular agent exhibits its control. So we have microbistatic agents, we have microbicidal agents, and we have microlytic agents. If those agents are specifically aimed at bacteria, we call them bacteriostatic or bactericidal or bacteriolytic, virucidal, fungicidal, right? All of those different things, but a catch-all phrase would be to call them microbistatic, microbicidal, or microbilytic. So the prefix there gives you an idea of who you're controlling. The end of the word, the suffix there, gives you an idea of how you're controlling them. So all static agents, if you think about the word static, stasis, staying the same, all static agents control microbial growth by inhibiting binary fission, and we can see what happens when a static agent is added to a growing culture by looking at this graph on the left-hand side of the slide. Notice we have two measures of growth. One is a measure of the viable cell count. The other is a measure of the total cell count. Remember, we talked about those in the previous unit. Total cell count includes live and dead cells. Viable cell counts, which we would record as CFUs per mil, tell us only those cells that are alive and capable of binary fission. So it looks like we've got two different kinds of measurements here. Maybe the viable cell count is a result of serial dilution and spread plating, and the total cell count is a result of spectrophotometric results. But if you watch both of those, they increase over time. So we're moving through time and increasing in cell number over time. And when this static agent is added, notice both the viable cell count and the total cell count have a slope of 0. So there's no change in the cell number over time. So you have stopped binary fission. You have assumed stasis here, and so we call that a microbistatic agent. If we move over to the middle graph, we have kind of a different situation here where we look at the total cell count. It's increasing over time with the addition of the microbicidal agent. There is a slope of 0. There's no change in the total number of cells over time, but this will highlight for you how important it is to have maybe those tedious and time-consuming processes which give you a viable cell count, because notice when you add the microbicidal agent, though the total cell count stays the same, the viable cell count drastically drops over time, has a very steep negative slope, and so you're killing those organisms. So this is a microbicidal agent. And then finally on the right-hand side, we have a microbilytic agent. So whereas static agents cause stasis of the population and cidal agents kill the population, lytic agents lyse the members of the population. So at the addition of the microbilytic agent, not only do the cells die, but the drastic reduction in the total cell count tells us that all of those cells die because they're being lysed. So as we're looking at different disinfectants and antiseptics and different methods of control, we want to be considering whether or not they are static, cidal, or lytic agents. Before we start talking about the methods of microbial control, I want to give you a couple of terms. The first one is really important because it allows us to compare all different methods of control for their effectivity, and that is something called the decimal reduction time, or DRT. And the decimal reduction time is the time it takes for a given control method to reduce the cells in the population to 1/10 of what they were. So if we plot these on a logarithmic scale, notice the decimal reduction time there on the blue line is the time it takes for that population to go from 100% to only 10% surviving. And that, in this case, takes 40 minutes for that blue line, and we call that the decimal reduction time. So in this graph, we can compare the decimal reduction time of 3 temperatures, but the utility of the decimal reduction time is that we could also plot a chemical that's being used as a control agent, gamma rays or ultraviolet light or anything like that, and we could compare that time frame between each of those methods of control and determine which of the methods of control is best. But here we're just looking at 3 temperatures. Nevertheless, we can look at those temperatures, and as you might suggest, the higher the temperature, the better it is at controlling microbial growth. For the culture that's being exposed to a high temperature of 50 degrees, the decimal reduction time is 40 minutes. For the population who's being exposed to 60 degrees, the decimal reduction time is about 12 minutes. And for the population that's being exposed to 70 degrees, the decimal reduction time is about 3 minutes. So as we would expect in this case, because they're all temperatures, the higher the temperature, the faster the decimal reduction time. When we're talking about temperature, though, and that's why this is used in this diagram here, is we can talk about different aspects of the control as well, and those would include the thermal death point and the thermal death time. So notice both of these, we've added thermal to our description, so we're talking specifically now about methods of control that employ heat. So the thermal death point is the lowest temperature at which all cells are killed within 10 minutes, and the thermal death time is the time it takes to kill all cells at a given temperature. So you might think about the thermal death point being useful if you were to say to yourself, okay, I have 10 minutes, that's all I have is 10 minutes. How hot do I have to get this thing in order to kill all of the microorganisms? And for thermal death time, you might think, okay, I don't have limitless temperature. I have only 50 degrees. I just need to know how long I have to cook this thing at 50 degrees in order to sterilize it. So you can use both of these pieces of information in the lab to control microbial growth. And unfortunately, for thermal death time, with these three temperatures, we're unable to determine the amount of time it takes to kill microbes at all of the temperatures, because notice at 50 degrees, we do not have a point in time where the culture has been sterilized. However, we can determine the thermal death time for both 60 degrees and 70 degrees, because that's simply the point at which the line crosses the x-axis. So at 60 degrees Celsius, the thermal death time would be 40 minutes, and at 70 degrees Celsius, the thermal death time would be 10 minutes. We can also look at these data and try to determine the thermal death point. So for that, we'd have to look at the x-axis and find the 10-minute mark and see if either of these temperatures was able to kill all microorganisms within 10 minutes, and sure enough, 70 degrees is our thermal death point. So notice the thermal death point is expressed as a temperature. The thermal death time is expressed as a time, but it's very important that you include the temperature. So the thermal death point is 70 degrees. The thermal death time at 70 degrees is 10 minutes. The thermal death time at 60 degrees is 40 minutes. And the thermal death time at 50 degrees cannot be determined from the data that you have in front of you. So that's a little bit of terminology before we move into this idea of controlling microbial growth. One more consideration to controlling microbial growth is various things either within the microorganism or in the environment that will change the thermal death point or the thermal death time or the decimal reduction time of a given organism. So they will make the decimal reduction time longer than it would otherwise be. So in this case, it would mean that at 50 degrees, it takes more than 40 minutes, or at 60 degrees, it takes more than 12 minutes, or at 70 degrees, it takes more than 3 minutes. So what are the things that will increase the decimal reduction time and by that decrease the treatment effectivity? The first of those is whether or not you have a ton of microorganisms present. When you have a huge load of microbes, you have a lot of microbes in a given area, it's going to take longer for you to kill them. The decimal reduction time will be increased. And the reason for that is because those microorganisms on the outside of the culture will be exposed first and will kind of buffer and protect those on the inside. So you have to have enough time for all of the microorganisms to be exposed to your method of control, right? If you have broth that you're trying to heat enough to kill all of the microbes inside, you have to apply that heat for long enough that it actually penetrates and increases the temperature of the entire flask. So when you have a high population load, as you can see from this graph here, it will take longer for you to kill all those microorganisms as opposed to if you had a low population load. So fewer microorganisms are easier to kill than many microorganisms. And that's something that if we were in the lab together, we would be employing for routine disinfection of what we assume to be a clean countertop. We spray a disinfectant and we wipe that disinfectant off. If you were to spill microorganism onto the counter, now we have a higher microbial load in that area on the countertop. So I would spray it and then allow the disinfectant to sit there so that the disinfectant would have time to penetrate the entire load of microorganisms and kill all of them. So the number of microorganisms, as it increases, the length of exposure to any treatment must also increase, or you won't control the growth of the microbes adequately. The second consideration is actually what's going on in the microorganism itself. If the microorganism has a glycocalyx, if that glycocalyx is highly arranged into a capsule, that can be difficult for methods of control to penetrate. The mycolic acid layer of acid-fast bacteria, the lipopolysaccharide outer membrane of gram-negative bacteria, the cell wall can make it difficult. Of course, spores will make it nearly impossible for some things to penetrate into the cell and cause damage. So you have to consider what it is about the microorganism that might be affecting your ability to control their growth. And that's why we set all of our disinfectants and antiseptics and remember sterilization to sterilize the spore, because the spore is the microbial characteristic that will allow it to survive for the longest period of time. Switching back to an environmental concern, we have moisture, and moist environments tend to increase treatment effectivity, while dry environments tend to decrease treatment effectivity. As you can see from the graph here on your slide, we have two cultures. One is wet, one is lyophilized, which means it is dry. And if we look at those over time, the solid line, the solid red line, which is the wet culture, so in broth, reduces very quickly, very sharp negative slope. So whatever this treatment is, it's working very well. Whereas the lyophilized culture doesn't change over time, no change in the cell number over time, which means that this is not a useful method of control for this dry or lyophilized culture. Now, in this particular experiment, it's temperature that's being applied to both the wet and the lyophilized or dry culture. And the reason that temperature doesn't work can be seen in the dotted lines. If you look at the red dotted line, the temperature of that wet culture very quickly increases and causes the death of the microorganisms, whereas in the lyophilized culture, notice the temperature does not change over time. And so the reason this dry culture is not affected by the temperature is because air is a very poor conductor of heat. And so the heat actually can't get into the culture. Nobody's being exposed to that temperature, and so nobody's dying. Whereas water is very good at conducting heat, and so the water conducts the heat into the entire culture and can very quickly sterilize the culture because of the moist environment. And that's something that we take to heart when we talk about sterilizing things. We typically in the lab sterilize using something called an autoclave, which we'll see in a few moments, but one of the marks of the autoclave is moisture, because we know moisture increases treatment effectivity. Another environmental consideration is the amount of organic matter that the microbes are surrounded by. Similarly to the fact that microbes can protect other microbes by absorbing the heat or the chemical or whatever it is that you're using as a method of control. Dirt or fecal material or things like that can also absorb the heat and absorb the chemicals and shield the microbes from the treatment. So organic matter in the environment will protect microorganisms, which is a lot of the reason why we clean wounds before we dress them and clean them before we apply antibiotics or antiseptics, because we need to remove that organic matter so that the treatment can get right to the cells and be as effective as possible. One final consideration when you're talking about treatment effectivity is something I mentioned in the previous lecture, and that is the ability of a microorganism to produce a biofilm. And so a biofilm, as you remember, is something that's created by microorganisms that almost allows them to act like a multicellular organism. They land on a surface and then they begin to secrete this EPS, and then they start developing a biofilm, and the biofilm becomes this irreversibly attached structure from which bacteria can be released and within which bacteria are protected from all sorts of things like antibiotics, and it makes them very difficult to treat in patient populations where they are typically referred to as vegetations, not biofilms. So in the medical setting, you won't typically hear biofilm, you'll hear vegetation. And just to give you an example of a vegetation, I gave you one of Staph aureus on a catheter. In the previous lecture here, I'm showing you an echocardiogram of a vegetation on the mitral valve of this patient. As you can see, the vegetation, which is this biofilm filled with microorganisms, is spanning nearly the entire width of the left atrium, making it very difficult for this patient's blood to move through the heart. And just as a way of looking at that clinically, I want to show you some data from an experiment where they exposed different kinds of cells that were capable of making biofilms to two different antibiotics. So we have tobramycin on the left and ciprofloxacin on the right, and we've exposed these cells to these antibiotics, and let's check out what happens. So if we look at tobramycin, we have both control and experimental data. The open circles and triangles and squares are the controls, and the filled in circles, triangles, and squares are the treatments. So whether you have planktonic cells, which are not capable of producing a biofilm, or cells that are planktonic but are capable of forming a biofilm, or you have a biofilm colony, when there is no antibiotic given, notice all of them survive over time. There's no reduction in the number of cells over time, and in fact, they are increasing a little bit over time. When you add the antibiotic, notice that both the planktonic cells and the biofilm cells are very sensitive to this antibiotic. They very quickly start to die. And so what we have here are cells that are sensitive to this antibiotic tobramycin. But once those cells are allowed the time to make a biofilm, which doesn't take much time at all, remember from your biofilm enhancement assignment, right, happens very quickly. Once they are within that protective biofilm, notice that the biofilm colony, the filled in squares, follows just about identically the track of the controls, which says they're no longer sensitive to this antibiotic. So even though in the lab we would report that organism as sensitive, in the patient it would not help to fight that infection at all. And a similar thing happens with ciprofloxacin. You have the biofilm colony, the squares that are filled in, growing at just about the same rate as the unexposed cells. So the biofilm protects these cells from antibiotics. it likewise protects the cells from your immune system. Your immune system comes in and it can't get in and bust through that biofilm to get the cells inside. So the benefits to an organism of producing a biofilm are huge. And the consideration from our perspective is that we have to figure out a way of getting rid of the biofilm so that we can actually treat effectively the patients that are infected with the organism that's capable of producing the biofilm. All right, so finally, we get to talk about the kinds of treatments that we can use. And we're going to start first talking about physical methods of control. And the first of those is heat, which we've kind of been alluding to already. So heat can be used, as you know from the previous lecture, at high temperatures. Proteins denature and cells thermally lyse. So heat can be used to control microbes by killing them, and low temperature can be used to control microbes by slowing their growth. And we talk about two kinds of heat. We talk about dry heat and we talk about moist heat. Dry heat kills microorganisms by completely oxidizing them, so basically turning them to ash, kind of like if you took a log and put it in a fire and in your fire pit and came back in the morning and there was ash there. That's basically what we do when we use dry heat to incinerate our microorganisms. And we do that routinely in the microbiology lab by using an Bunsen burner, where we take the loop and we put it into the flame of the Bunsen burner until it's red hot, at which point it will have incinerated anything that's on the inoculating loop and sterilize it for you. The other thing that you can do is use hot air sterilization, which is basically like putting things in an oven and baking them. The problem with hot air sterilization is that it is dry, and as we know, air does not conduct heat very well, so it takes a really long time for hot air to be useful as a method of microbial control. And just as a comparison, hot air, if you want to sterilize something using hot air, you have to heat it to 170 degrees Celsius for 2 hours. If you utilize moist heat, you only have to heat it to 121 degrees for 15 minutes. So it's a much faster and lower temperature that's required in order to sterilize with moist heat. And moist heat works by denaturing proteins and popping spores, and our most common way of using moist heat for sterilization is a machine called an autoclave, which is used in microbiological labs to sterilize media prior to its use and also to sterilize media that organisms have been grown in before we dispose of them. And it utilizes pressurized steam in order to sterilize whatever is inside of it and also to pop spores. So autoclaves are a method of moist heat sterilization which denatures proteins. Now, one kind of heat control that's used that does not achieve sterilization but is very commonly used commercially is something called pasteurization, which, as its name suggests, was discovered by Louis Pasteur. It's important to note this does not sterilize, it sanitizes food items. It lowers the number of microbes to a safe level for consumption. And historically, pasteurization was done at 63 degrees Celsius for about 30 minutes, but we've learned that equivalent treatments are 72 degrees for 15 seconds or more like a flash of 135 degrees Celsius for less than a second. And those are really important because cooking something at 63 degrees Celsius for 30 minutes can drastically change the flavor, and we're talking about foods. So just as a comparison, we have these different kinds of pasteurization that are used today. HTST, which stands for high temperature short time or flash pasteurization, is that milk being heated at 72 degrees for 15 seconds and then bottled and refrigerated, right? Important to refrigerate it because 72 degrees for 15 seconds will lower the number of microbes, but it will not kill them completely. It doesn't sterilize the milk. And so we have to employ low temperature in order to control whatever microbes are left and keep that food from spoiling. as opposed to ultra high temperature pasteurization. So if we take that milk and we heat it at 138 degrees Celsius for 2 or more seconds and seal it in an airtight container, that can actually be stored for up to 90 days without refrigeration because you've sterilized the milk. So pasteurization is a way of employing heat to control the growth of microbes to the point that the food is sanitary, and now today to the point that the food has been sterilized. So heat is the first of the physical methods of control. The second method of physical control is salt or sugar. Any molecule that causes osmotic stress and causes water to leave the microorganism will desiccate or dry out the microorganism, remove all of the water from inside it, and the microorganism will be incapable of growing. And this is historically a very important method of control. And there's a couple of different foods shown here: raisins, of course, beef jerky, salted fish, and then in jellies and jams, it's the sugar that keeps microorganisms from growing. So people for thousands of years have understood that if you dry out food or salt or sugar it heavily, that food will last and will not be spoiled by microorganisms. So salting and sugaring causing osmotic stress is another physical method of control. So we have heat, osmotic stress, actually the physical removal of microorganisms using something called a filter. So the filter can actually trap microorganisms and allow what flows through the filter to be sterile. So think of this kind of like if you're cooking pasta and then you run the pasta through a colander. When you're done cooking it, right, the water flows through and all the pasta gets trapped. So in this thought experiment, the pasta are the microorganisms, and then what goes through would be the sterile filtrate. So you pour in a sample that could be contaminated, you pass it through a membrane filter, and you collect a sterile filtrate at the bottom. And that can be done on a larger scale, like you see here, where you might be able to filter sterilize, a liter of fluid at a time, which might be important to you for time consideration, but also is routinely used in the lab when you're working with something that is heat labile, like an antibiotic. If we were to put an antibiotic into the autoclave and use heat to sterilize a broth containing an antibiotic, it would actually break apart the antibiotic and render it useless. And so we call antibiotics heat labile. They would be damaged by heat. And when something can be damaged by heat, you don't want to use heat to sterilize it, so you might use filter sterilization. You might also use filter sterilization, as I mentioned, because you don't have a lot of time. You don't have that 15 minutes to devote to putting it into the autoclave and then the time it would take for that material to cool down from 120 degrees Celsius to a temperature that you could work with it. So you might use it just to do something more quickly, and you might need only a little bit. So we also have these very tiny membrane filters where you can draw up material in a syringe, add one of these small sterile filters, and then push using the syringe the material through. The organism will become trapped in the filter, and what will end up in the collection vessel is a sterile filtrate. This is commonly used not only in the lab, but also in the environment with HEPA filters. HEPA stands for high efficiency particulate air. So you're moving air through these filters and it's trapping microbes and other things inside them. And HEPA filters are very common. They might be in your car, you might have one in your vacuum at home. So as you're pulling air through into the cabin of your car or pulling air into your home through your vacuum, you're catching everything and then releasing clean air. So HEPA filters are very commonly used, and they're very good at trapping all sorts of things, including microorganisms that might be moving through the air, and so eliminating airborne pathogens that might cause disease. Our next physical method of control is radiation, specifically gamma radiation, X-rays, and ultraviolet radiation, which are higher energy than visible light, and so are capable of being absorbed and causing damage. Ultraviolet radiation causes what's called pyrimidine dimers. So ultraviolet radiation is actually absorbed by the DNA of the cell, and when you have adjacent thymine or pyrimidine nucleotides, it will cause a kink where they bind to one another, which ends up causing a deletion mutation when DNA polymerase comes through, because DNA polymerase will incorrectly read that dimer as one single thymine and add one single adenine opposite it. So this kind of damage caused by ultraviolet radiation damages DNA, and if you get enough of that damage, then you get death of a cell. Gamma rays and X-rays are actually so high energy that they ionize water, converting it to the free radicals that we talked about in the previous lecture. And of course, those free radicals will also attack and damage DNA, breaking it into small pieces. So the result of these methods of control are the same, damage to the DNA, but the damage is done very differently. Gamma rays and X-rays are said to cause ionizing damage. They are forms of ionizing radiation because they ionize water, and ultraviolet light is compared to those and is called non-ionizing radiation. So ionizing and non-ionizing radiation both cause damage to the DNA. Ultraviolet light directly causes damage to the DNA, whereas gamma rays and X-rays cause indirect damage to the DNA by first ionizing water to those reactive oxygen species or free radicals, which will then go in and damage the DNA. Both forms of radiation are very useful. Ultraviolet radiation is commonly used in hospital labs to make sure that the hoods within which we do our inoculations are sterile. So during the day, you will work in the hood because you're working with organisms that are at a BSL level that requires a hood. And when you're done working for the day, you spray down with a disinfectant, and then before you leave, you turn on an ultraviolet light, and you allow that ultraviolet light to shine on the surfaces of your hood so that when you come in the morning, it's been sterilized. Gamma radiation and X-ray radiation is used in order to sterilize foods. These are some apples that have gone through a gamma radiation treatment in order to keep them from spoiling. A lot of spices are radiated, and so both types of radiation are used routinely. All right, so that wraps up the physical methods of control. So we had heat, salt, and sugar. We had filters, and we had radiation as physical methods of control. Now, though we talk about different physical methods of control, we really are not going to talk about different methods of chemical control, because the way that chemicals work to control microbial growth are so varied that it would take too long to come up with just a couple. So instead of focusing on the kinds of control that chemicals exhibit, we're going to talk about the way we measure how well chemicals control the growth of microorganisms. And we're going to talk about two ways in particular. The first is shown here, and that is something called the use dilution test. And the use dilution test is helpful because it will identify for you whether or not a given chemical is bactericidal. And the way that that's done is by taking a metal ring, which is shown here, and dipping it into a bacterial culture and allowing it to dry, as you can see here, at 37 degrees Celsius. Then that ring that has the dried organisms attached to it is placed in the disinfectant for 10 minutes at 20 degrees Celsius, and then it's removed and put into broth without disinfectant in order to see whether it survived the disinfection for 10 minutes. So if it survived, then this agent is not bactericidal. If it doesn't survive, the agent was bactericidal. The next evaluative method that we're going to talk about for chemical control of growth utilizes something called a disk diffusion test. And a disk diffusion test you've seen in lab. A disk diffusion test allows you to apply a given chemical agent to the surface of agar that's been impregnated or seeded with microorganisms in order to see whether those organisms are inhibited from growing. And the inhibition of that growth can be seen in something called a zone of inhibition. So in this diagram on the left-hand side, you have a plate that has been seeded with microorganism, and these antimicrobial discs have been applied to the surface of it. The microorganism grows up to a point, and then it becomes inhibited by those chemicals, and you see that as a zone of inhibition. So notice on the left-hand side, we have two organisms, Pseudomonas aeruginosa and Staphylococcus aureus. Pseudomonas aeruginosa only has a zone of inhibition around one of those chemicals, and it's very small, whereas Staphylococcus aureus has a zone of inhibition around all four of the chemicals, and they're very large. So we would say that Staphylococcus aureus is much more inhibited by these chemical agents than is Pseudomonas aeruginosa. So the disc diffusion method is a measurement of chemical control that will tell you whether or not a given chemical is bacteriostatic. It will tell you whether or not it inhibits it from growing. And again, the procedure begins with a plate seeded with organism, and then filter papers which have been soaked with either a disinfectant or an antibiotic or some chemical agent will be placed on that seeded agar, and then the plate is incubated and examined for growth. So the plate contains the antibiotic or the disinfectant or the chemical, whatever the agent is, and the cell is never allowed to grow outside of the presence of that agent, and that's why this will only tell you that it's bacteriostatic. Within that zone of inhibition, which is either caused by a disinfectant like chlorine or O-phenylphenol or quaternary ammonia compounds or hexachlorophene, or an antibiotic like Optochin, only tells you that the cells within that zone have been inhibited from growing. They could be alive, it's just that they haven't been able to grow into visible colonies. So that's why this is a bacteriostatic test. So the use dilution test will tell you whether or not something is bactericidal. The disc diffusion method will tell you whether or not something is bacteriostatic. As we wrap up this lecture, I'd like to just talk about microbial control in terms of how easy or difficult it is to control microbial growth. So not talking about whether something is microbicidal or static or lytic, but just talking about how given agents are killed and how easy they are to kill. So the most resistant microbes to chemical and physical disinfection are prions. I would say, in contrast to this diagram that's found in the Tortora textbook, that non-enveloped viruses, because they're surrounded by protein, are up there with, not with the prions, but up at the top there between the prions and the endospores of bacteria, followed by the acid-fast bacteria, protozoan cysts, protozoa, gram-negative bacteria, fungi, gram-positive bacteria, viruses with lipid envelopes, and then at the very bottom there is you. Your cells are the least resistant to all of these methods of control. And incidentally, that's why the viruses with lipid envelopes end up way at the bottom here, because viruses with lipid envelopes function very much like your cells. That lipid bilayer membrane is the only protection they have from the outside world, and that lipid bilayer membrane can be very easily dissolved just with washing your hands with soap and water. So you're the biggest wuss, then the enveloped viruses, then the gram-positive, because they only have that peptidoglycan cell wall, then the fungi, who do have a cell wall and can exist in areas where osmotic stresses are really high. Then we have the gram-negative bacteria, because in addition to that little bit of peptidoglycan, they have the lipopolysaccharide outer membrane. Then we have the protozoans, the cysts of protozoa, which are much like the endospores of bacteria. Then we have the mycobacteria, which have that thick peptidoglycan and the mycolic acid layer, which is very difficult to penetrate. The endospores of bacteria, which of course are very difficult to penetrate and are highly dehydrated, and so are not as susceptible to ionizing radiation, then viruses without envelopes, and then finally prions. But the good news is that most of these things can be controlled, right? A person can retain their good health by washing these transient organisms off of their skin with soap and water. So the take-home is just to wash your hands with soap and water, and you will remove all of these, right? Because washing with soap and water removes them mechanically from your skin and protects you from the first step in infection, which is for them to just attach to you.